So we've been studying a process called autophagy, which is the cell's recycling mechanism. It's activated during times of stress to protect cells and to keep cells alive. It's a very good thing for the cell and indeed for all of us. It keeps us healthy throughout our lives. But within the lifetime of a cell, there is one point where autophagy actually must be inhibited, and that's during mitosis, when the cells divide, because the nuclear envelope breaks down and that can expose the genome to the autophagic machinery, which could actually chew up the DNA and cause damage to the DNA. So we've been trying to understand how autophagy is repressed during mitosis. In the other parts of the cell cycle, autophagy is normally repressed by an enzyme called mTOR. And we found that mTOR is actually inhibited during mitosis. It's inhibited by the master kinase regulator of mitosis called CDK1. And then we've shown that CDK1 actually takes over the functions of mTOR during mitosis to repress autophagy. The clinical implications of this are many forms of cancer are still treated with drugs which cause mitotic arrest. These are taxanes such as paclitaxel and so-called vinca alkaloids such as vincristine and vinblastine. These are drugs which disrupt mitosis and cause cells to stick in mitosis and then die. There is a possibility that if we can force autophagy to be reactivated when patients are undergoing treatment with those sort of chemotherapy drugs, that this may actually enhance tumour cell death by exacerbating genome damage caused by those drugs. And so one possible avenue to take this work forward would be to try and identify drugs which activate autophagy and to administer those together with paclitaxel or vincristine, drugs which are so-called anti-mitotics. The next steps are trying to understand at the molecular level how this process takes place, how this repression is achieved. We think we have a fairly good idea about that. We also need to understand if this applies to all aspects of mTOR signaling. So for example, mTOR exists in two complexes within the cell, mTORC1, which we have studied, and mTORC2. We need to understand if mTORC2 is also repressed during mitosis. We need to understand the molecular basis for this repression. And then with regards to autophagy activators, this is an area where we might be able to learn something from studies ongoing in neurodegenerative disease, where there is an aspiration to stimulate autophagy to clear misfolded and damaged proteins which cause neurodegeneration. So there are drug discovery efforts ongoing for diseases such as dementia to try and find drugs which activate autophagy. Can we take a lesson from those studies and actually develop our own drugs which will promote autophagy and specifically target the repression of autophagy during mitosis?